Very good afternoon to all. It's a very pleasure to be part of this HCCBS International Conference. Thanks for the Mizoram University. I have come from the Ministry of Ayush. I have to say that before going to the topic, just a nutshell, that today's world, nearly 70% of the population in the world are still depending on their traditional medicine system for their primary health care needs. All over the world, every country almost has their own traditional medicine system. For example, Chinese medicine system, African medicine system, Thai medicine system. Like that in India, we have also traditional medicine systems, Ayurveda, Siddha, Yoga, and Naturopathy. These are the things. In that, the major issue is the traditional medicine system, safety, efficacy, biological understanding for globalization is the major challenge. With this background, one of the medicines, Saraswata Grida, widely prescribed in the Ayurveda for the brain development or mental disorders, is being understood biologically or how it works, mode of action. That is the thing the presentation talked about. So next slide, please. Where is it? Saraswata Grida is a classical Ayurvedic medicine. It is being practiced nearly 5,000 years in India for the mental disorders. The key is a base, it is an internal medicine. Whenever we talk about this Saraswata Grida, the present day context, the depression is the major issue. In the depression, 80% of depression leads to Alzheimer's disease. That's what the data says, that nearly 5% of men and 6% of women who are above 60 years are going to be affected enormously by 2050 due to Alzheimer's disease. So for the Alzheimer's disease condition patients, the Ayurveda medicine Saraswata Grida, when it is being given, how it works, how far the improvement is the study. Here, this is a rat experimental preclinical study to understand the mode of action of Saraswata Grida. Okay. So for that, before going for the efficacy, naturally the toxicity study should be done for the safety purpose to understand the safety of the drug. It has been conducted. The another objective is efficacy. So the trial drug was prepared in our National Ayurveda Research Institute, Panchakarma. Quality control analysis was done. Toxicity studies have been carried out. Then efficacy study. So this is the Saraswata Grida. It contains 10 plant ingredients. Of course, Piper nigrum, Piper longum, Gingiber officinae. These are all things. The preparation is as per the Ayurvedic formulary of India. There is standard method. There is a pharmacopoeia, Ayurvedic pharmacopoeia of India, AP. It has to adhere all the guidelines. The physicochemical parameters will be done for the raw materials or finished product. Toxins and mineral oil will be assessed in raw material finished product. Microbiological parameters also assessed raw materials and finished product. After these are all the things. Once ensures the quality of the preparation medicine, then it is being given to the animals for the toxicity study. Means the safety of the Saraswata Grida in animals. As per the OECD 423, acute toxicity study, repeated dose 28-day oral toxicity as per OECD 407. As per 407 has been carried out. Both the studies showed the trial drug is safe. LD50 is greater than 5,000 milligram. This is the 1,000 milligram per dosage. The efficacy study where the aluminum chloride 300 milligram per kilogram body weight has been given to the experimental animals, rats, visceral albino rats. And the treatment group, disease control group, average, low, medium, high dose have been given. Standard drug rivastigmine was used. This is a 1-month study trial drug. So after that, various biochemical markers, biomarkers for with reference to Alzheimer's disease was measured, including BDNF, rat acetylcholinesterase, TNF alpha, vitamin C, antioxidant histopathological studies have been carried out. The study showed that the study, acute study level, it is a greater than 2,000 milligram per kilogram, means you can go up to the 5,000, that is the range. It is a safe up to 2,000 milligram. OECD 407 28-day study proved safety of the Saraswata Grida. The pharmacological parameters have been carried out for how far the behavioral assays have been improved while using the Saraswata Grida. Here, this is the data I am presenting with reference to BDNF, acetylcholinesterase, tumor necrosis factor in serum samples. It showed that BDNF's level in disease control have been very low. It has been improved according to the dose concentration. Of course, the standard drug has their own level, rivastigmine. Similarly, acetylcholinesterase level have been decreased. our Saraswata Grada level tries to improve the acetylcholine esterase level. TNF alpha, the DC reference disease control, the 3 doses, the standard drug. Vitamin C, vitamin E level also have been influenced. Same way, the BDNF level and as well as acetylcholine esterase level have been measured in the brain tissues also. It also showed that disease control has a very 7.05 nanogram per gram tissue level, whereas it is trying to improve at the higher dose level, 10 medium dose level. Standard drug is 14.58. Acetylcholine esterase level is very increased in disease control level, disease control group, whereas the treatment of Saraswata Grada trying to decrease the acetylcholine esterase level. So acetylcholine level will be increased, acetylcholine esterase enzyme level will be decreased. This is the data. The histopathology also showed us cerebrum and as well as hippocampus region controlled as a treatment group, it showed improve. So here I wanted to say that the Saraswata Grada classical Ayurveda medicine, people used to say that how it works, whether it is safe or these are all issues preventing the globalization of Ayurveda. But even that medicine also works very well. We can understand through such type of biomarker assays. So safety was documented, efficacy of and as well as possible mode of action. It is a BDNF pathway, TNF alpha pathway. Antioxidant plays the role. Because of that, the Saraswata Grada works on Alzheimer's disease or mental disease. That's what the conclusion. So further studies always at molecular level has to be carried out for scientific understanding. Because we cannot go very individual molecule based study, because classical preparation or Ayurveda preparation, it is millions of compounds are present. We cannot elucidate structurally one compound to give the credit for entire activity. It will become the modern medicine one, or otherwise it will become the nutraceutical, or the cumulative effect, synergistic property of the classical traditional medicine system will be lost. So we will never work this one. For scientific curiosity, we can go up to the molecular level, how it may work like that. Here I conclude. Thanks for the acknowledgement my CCRS and who is the sponsoring for this project. Thank you. Thank you.
The next speaker, Dr. Sudesh N. Gaidan.
Dr. Selvam, in the meantime, next speaker is coming. So I would like to one question. What was the behavioral study?
Yeah.
Okay. Okay. Fine.
Thank you, sir. Yes. Good evening to all. I am Dr. Sudesh N. Gaidari. I am Assistant Director Pharmacology. I am from National Ayurveda Research Institute for Panchakarma, that is under the Center Council for Research in Ayurvedic Sciences. CCRS is one of the nodal agency for drug development and scientific validation of these Ayurvedic drugs under Ministry of Ayush. We are having the different centers all over India. And this NARIP, that is Panchakarma Institute, that is in Kerala. We are having the excellent preclinical setup at our institute. We used to standardize drug, Ayurvedic drug, classical drugs, or coded drugs as per the regulatory guidelines. So like this, we have from last 10 to 20 years, our council has already been developed and patented many drugs also. So today, I am going to present the work of one of project allotted by my council. That is comparative analytic and integrative evaluation of selective medical plants through experimental pharmacology. So this is not the simple comparison with the two medicinal plants. It has been, this council and the Ministry of Ayush has already been developed the Ayurvedic pharmacopoeia, which contain the various standards of different single medicinal plants. Apart from this one, Ayurvedic Formulary of India, which contain the various formulations mentioned in the classical drugs, that has been mentioned in that Ayurvedic Formulary of India. And this formulary of this classical drug we used to revalidate, and apart from that one, we are just developing the new Ayurvedic drugs also. So as we are aware that this one in many herbal industries, they are using the many herbal medicinal plants for preparing the various Ayurvedic medicines. but they are not properly following the guidelines from preparation of medicines due to certain different problems also. So suppose this classical drug mentioned in the Ayurvedic pharmacopoeia needs the particular plant, but that is not available in South India. That is available as per the geographic location in North India, this one. So they are not using that particular plant mentioned in the Ayurvedic pharmacopoeia India. They are using the different plant available in the southern part of area. So this happens from last 15 years, this one in Kerala also, in other parts of India also. So to confirm the effect of that particular drug in that classical drug prepared by the pharmacy, to confirm that the efficacy of that drug, I have done this particular project and for the comparison. So as you are aware that this one in under Ayurveda, we are following these methods, the conceptual research, either retrieval and translation and digitization of the various manuscripts, and then it goes to drug research, that is for the drug standardization and preclinical study, and after that clinical trials. So under the drug research program, either you have to cultivate your own medicinal plants, or you can collect from the other sources, and then goes to pharmacology, and then drug standardization, chemistry, isolation if required, and then preclinical toxicology study and efficacy study. So, and these are the standards available in Ayurvedic pharmacopoeia India for classic for single drugs and compound formulations also, that we are following as per the guideline of Ayurvedic pharmacopoeia of India. And these are the different parameters. Apart from this one, we are doing for the heavy metal analysis, microbial loads also, fungal count, pesticide, and aflatoxin also shape light for all the single compound and compound formulation also. And this is the Ayurvedic pharmacopoeia of India. This one we have so far, we have published the single drug monograph of 640 plants in 9 volumes, and compound formulation 203 plants in 4 volumes. Apart from this one, Ayurvedic formulary of India, it contains classical formulation of 986 in 3 parts. So objective of my this particular presentation is to do the final phytochemical standardization as per the pharmacopoeia, Ayurvedic pharmacopoeia of India, and its safety evaluation through acute toxicity and efficacy study, that is analgesic and anti-inflammatory study of this all the source plant which are mentioned in the Ayurvedic pharmacopoeia and their substitute used in the particular area. So these are the 3 botanical source plant, Plumia, Manancellera. These are the source plant mentioned in Ayurveda, and in case of this 3 plant, most of this one alternative plant used in the in South India. So I have done systematic this pharmacognostical study also, microscopy of all these source plant, 3 source plants and 3 alternative plants also. So these are the pharmacology study of these all 6 plants. After that, we proceed for the standardization of drug, that is quality control of analysis of that on the 6 plants that we have done, and these are the different physicochemical parameters and preliminary phytochemical analysis of all 6 drugs. We have used for this one aqueous and hydroalcoholic extract. Ayurveda accept only the aqueous extract and hydroalcoholic extract, that is 50-50% water and ethanol only. Ayurveda not accept the methanolic or ethanolic or any acetone extract. So these are the result of this quality control of all 6 plants. So then acute toxicity of these all 3 drugs as per the OECD guideline 423, and for this one we have taken. These are the grouping and randomization of these all 6 plants. These are the results of acute toxicity studies, and result of acute toxicity is no mortality found up to the dose of 2000 mg per kg. And these are the efficacy study, that is analgesic and anti-inflammatory study. For analgesic activity, this 3 model has already been given: thermal harmonic by hot plate method, tail flick method, and paw pressure method. For chemical hyperanalgesia by formalin method, and anti-inflammatory activity by the carrageen-induced hind paw edema method. So these are the different models we have conducted analgesic study. Anti-inflammatory by carrageen-induced acute inflammation, and these are the study design. These are the different groups and with standard control also. So result of efficacy studies, these in both the doses, this on the analgesic activity has already been highly significant at 60 minutes of the post drug administration as compared to control. Like this, these are the results in the tabular and graphical representations. And the substitute plant produces the equivalent analgesic and anti-inflammatory potential similar to the source plant mentioned in the API. So we can justify the treatment of acute and pre-inflammatory condition where the source plant are endangered and not available can be justified. So thank you.
Thanks, sir. Thank you so much, sir. The next speaker, Kirti, anti-obesity potential of seaweeds. Absent. So then, Chandan Kumar Ghosh, Kurkumi.
Okay, thank you, organizer, for giving me the opportunity to present here. That topic or the discussion that we have selected here, that is a curcumin nanoformulation to treat the triple negative breast cancer cell. As we are running short, so I will try to summarize in a quick time. So layout of my talk is first some introduction, then methodology, results, and fundamental understanding, and lastly I should conclude somewhere. So as we know, so I am skipping all these things. What is the definition of cancers? What are the correlations of the cancer cells? So I am skipping all these things. So particularly, so these are the different classification method of the cancer. Now, out of this, we are particularly concentrated on the breast cancer cell model. This is the highest among the women, in fact, throughout the world. In fact, in India also. Now, these people, breast cancer, they can be classified into 4 categories. In the estrogen positive, progesterone positive. So these are hormone driven. The third one is the HR2. This is so first two is the protein driven. Third one is the hormone driven. But accidentally, the fourth one, where all these that hormone or protein, they are absent. So they are defined as the triple negative breast cancer. And most interestingly, it is found almost 10 to 15% among the all breast cancer patients. So out of these, it will be very difficult to treat the breast cancer, triple negative breast cancer, because there is no definite receptor for this. So as we are almost all other cancer model, we are going to do the chemotherapy. Then these are the main drawbacks to treat the to use the chemotherapy to treat the triple negative breast cancer. So what may be the other alternative? The targeted therapy may be another alternative for this purpose. So in targeted therapy, more than 2 or 3 or different therapeutic models, therapeutic activity, they are combined into a single platform to treat the breast cancer cell model. So basically here one particle, they are used as a drug carrier, and then this particle or drug carrier may be of different type, maybe polymer based or inorganic nanoparticle based, organic nanoparticle based. So there are lots of variation in the drug delivery system, but at the same time, when these drug carrier are being developed, the drug has to be developed or precipitated to some the target positions. So there are 2 different mechanisms for these drug releasing mechanism. One is the active targeting, where some receptor is needed, because there in TNBC there is no definite receptor. So there is another targeting mechanism is a pH driven. So what happens in compared to normal cell, the cancer cell, it is highly acidic in nature. So that acid environment can be used to deliver the drug delivery at the proper places. So in this context, the nanomaterials plays a significant role, where not only the therapeutic purpose can be served, but using the same nanoparticle, the diagnostic purpose can also be created. So for this type of system, the therapy as well as diagnostics both are possible. So again, the diagnostics mechanism can be divided into 2 categories. One is anatomic based and second was the molecular based. Among this one, we are just concentrating on the MRI part, because as we are choosing the nanoparticles of, as I am going to discuss, we have chosen the Prussian blue nanoparticle for this purpose. As we know, that Prussian blue is it is of magnetic character, so magnetic nanoparticles, they are used for this MRI purpose. So other advantage of these Prussian blues is that it can be used as a photothermal and photodynamic therapy, as well as it can be used the MRI in both T1 and T2. So these are the two different imaging protocol in MRI. So this T1 will make brighter field more brighter, and T2 it will make the darker field more darker. So it will create more contrast efficiency in the diagnostics. And since these nanoparticle Prussian blue, it is highly porous in character, so it can be used to deliver a drug to a high extent. So again, this Prussian blue is an FDA approved nanoparticle. Now, this nanoparticle, it has a different size, and of course, from our previous experience, we have observed that different size, they have different efficacy. So we have chosen just reflex model, a reflex system, reflex method to synthesis all the Prussian blue nanoparticles. So these are the beautiful image of the Prussian blue nanocubes, how they have, those nanoparticle or nanocubes we have obtained. Again, bare nanoparticle we cannot use. We need some biocompatibility. Normally PEG has been used for this purpose. And again, the drug, chemotherapy drug, as we are choosing here, it's a curcumin. Curcumin, it is a natural flavonoid that is being used. As we are talking about the sustainability. So here the curcumin as a natural flavonoids play a great role. And for targeting, it has been achieved by the hyaluronic acid. So these are the all characterization mechanism, the FTIR, UV visible, as well as FESM, respect to FESM images to confirm all these functionalization as well as loading. Now we have gone through the MRI images, different functionalization stages. We have performed the MRI images. So ultimately, these are the two parameters that we have got of the final product of two different size nanoparticles. This is the best one. And if we compare with the standard, commercially used nanoparticle, that is the GT based, it has a high R1 value, it is 3.2. But in our case, it is 6.12. That is almost double in comparison to the commercial nanoparticle used for the MRI contrast efficiency. So again, as I mentioned, that all this functionalization process, they have a different impact on this MRI activity. So we have tried some in vitro in theory to explain this functionalization process and also the importance. Actually, all these parameters say significantly affect this R1 and R2 value. We are not going into details into that mechanism, but again, we have optimized all these parameters to find out the best result MRI as a MRI contrast agent. So next, we are talking about the photothermal therapeutic activity, that is PTT activity out of this. So here, some laser light basically use of 808 nanometer. It is used to increase the temperature, local temperature. So by heat treatment, by inducing the heat, the cancer cell can be treated. So these are some images of how the temperature it is going to increase. So this is the repetitions of the cycle. So again, these are some effect that the drug release kinetics at the different pH condition. This is the acidic condition and this is the neutral condition. So the drug release profile, it is achieved here in the acidic mode. So again, we have carried out the photodynamic therapeutic activity, that is mostly the singlet oxygen generation out of this 808 nanometer. So our nanoparticles, so we just started the therapeutic activity of the system. Here, this is the therapeutic activity of the only drug, and this is the therapeutic activity in the presence of light also. So there's a significant changes in that therapeutic activity. And again, if we compare the IC50 value, it is only 14.72. At a standard chemotherapeutic drug, it is almost 30 or 50 microgram per ml. So these are some different assays. So what is the mechanism that we have also tried to investigate here? So these are the different protein mechanisms that we have identified after the bioinformatics studies. Then we have also tried that what is the other fundamental mechanism, and we have confirmed that this is the inflammasome-mediated pyroptosis pathway that is going to kill the cancer cell. So what we can conclude is that MRI dependence in the size and all these spheres, their natural flavonoid is a curcumin, it can be used as a chemotherapy drug instead of synthetic drug. And here we are getting the pyroptosis mechanism. Again, in the literature, and also we are currently working on this pyroptosis mechanism. It is going to impact the immunotherapeutic activity of the cell. So it will be very interesting in future to incorporate the immunotherapeutic activity within our system. So I should, for this work, I should acknowledge our funding agency, and then thank you.
Thank you. Thank you, sir.
Thank you.
The next speaker, Roland Yamtham. Yeah. Human health risk assessment through consumption of chromium lead and nickel exposed vegetables, Nagaland, Northeast India.
A very good evening to each and every one of you. I am Roland Yantan from Mizoram University, from the Department of Life Science and Bioinformatics. And today I'll be discussing on human health risk assessment through consumption of vegetables exposed to chromium, lead and nickel in Nagaland, Northeast India. So as we all know that pollution towards the environment, it has become a huge problematic thing and remains a great threat. So there is a great concern among the developed countries going around globally. So one of such concern which I'm going to discuss here is contamination of these vegetables by the heavy metals. So what are these heavy metals? These heavy metals are non-essential elements and non-biodegradable elements which tends to persist in the environment for a long period of time, which can be very toxic. And this metal, how does it get into the agriculture ecosystem is with rapid development and increase in urbanization and increase in population growth, there has been a major increase in the demand for food. So to meet the demand for the food, lots of pressure like intensive agriculture or overuse of agrochemicals like pesticide fertilizers are being used, which is affecting to the agriculture ecosystem. And once you consume these exposed vegetables by the heavy metal, in the long period of time, they can be a very fatal and have a negative impact towards a human health, such as they can affect the such organ like oral diseases, blood diseases, and gastrointestinal diseases, and all the non-cancerous and cancerous diseases. And these are the methodology, and here I'll be discussing on the concentration of the heavy metal on the vegetables and a special distribution of this concentration of vegetable by using ArcGIS, and also the non-carcinogenic and carcinogenic risk assessment using the health risk assessment model. And these are some of the parameters which are being used in evaluating the non-carcinogenic and carcinogenic health risks. And this is the study area I have conducted in the Govt districts, which is one of the districts in Nagaland. And so this is the results. Next is the result in discussion. So what I found is in all the three metals have shown to have a higher limit which have been set by the USPA and WHO and FAO. So it shows that it is the vegetables are contaminated. And this is the special distribution using the Cricken interpolation method by ArcGIS, which shows that mostly as for the lead, it is showing that most of the vegetables are being concentrated in the eastern and southern part. And for the chromium and nickel, it is mainly concentrated in the eastern, and little bit is showing on the northern and southern part of the region. And this is the chronic tele intake of the vegetables by both the adult and the child, which are used for the non-carcinogenic and carcinogenic risk assessment. And for the non-carcinogenic risk assessment, as we can see that if the HI value is showing to be above 1, then it is considered that it is likely to have this health risk effects. Whereas if the HI value is shown to be below 1, then it is unlikely to show any health effects. So as in my studies, I found that it is both the adult and the child have shown that it is showing way above the limit, and chromium remains to be the highest contributor. And when we see the chart, when we compare it between the adult and the child, in the non-carcinogenic risk assessment, the child has shown to have a greater risk of this health and carcinogenic risk assessment. As for the carcinogenic risk assessment, if the TCR, that is a total carcinogenic risk assessment, if it shows 1 into 10 to the power minus 4, if it is greater than this, then it is non-acceptable. And if it is showing lesser, then it shows that it can have a highly significant health risks. So when we see even for the carcinogenic risks, okay. So in conclusion to my studies, vegetables were found to be concentrated, and when for the carcinogenic, for the health risk assessment, both the adult and the child was showing to have both non-carcinogenic and carcinogenic risk assessment. So like from this study, there is a need of proper policy management, remediation, research needs to be taken so that these health risks need to be reduced and also to safeguard the vegetable. So thank you.
Thank you so much.
Good afternoon, everyone. The topic of my today's talk is mitochondrial genome alteration and their associated risk towards breast cancer among high-risk tribal population of Northeast India. A brief background and rationale of the study. It's breast cancer is a malignant tumor that often leads to the death of women worldwide, affecting diverse socio-economic classes. Despite the established contribution of nuclear genes in breast cancer, the significance of mitochondrial DNA mutation as appropriate features in breast cancer formation has been recognized just decades ago. Mitochondria are the major source and target of intracellular reactive oxygen species, and they play a very important role in breast carcinogenesis. They also facilitate intrinsic apoptotic pathway, and mutation in ATP6 genes leads to a lower apoptotic process. Several studies have reported a large number of mitochondrial DNA variations in breast tumor samples, which suggests that mitochondrial DNA mutation plays a significant role in breast carcinogenesis, and several mutations have been linked to breast cancer in several populations. However, these findings remain inconsistent because some variants which has been associated with breast cancer in certain populations shows no association in yet other population. Consumption of tobacco in different forms is also known to elevate ROS production, causing mitochondrial DNA mutations. So with the consistent increase in breast cancer cases and the fact that Mizoram has a certain unique lifestyle habits, such as consumption of tobacco-related products in different forms, it is very important to find the underlying genetic risk factors among this population. So with all this background, this study focused on evaluating the role of mitochondrial DNA mutation and their contributing environmental risk factors towards breast cancer development. This is a global scenario of breast cancer in terms of incidence and mortality. Breast cancer ranks the second highest in terms of incidence, and mortality rate ranks the 4th position. And in case of Mizoram, it's the 3rd highest position following cervical cancer and lung cancer. And moving on, I have a total of 44 samples, 32 breast cancer cases and 12 healthy controls. The subjects were interviewed based on a structured questionnaire, and blood was drawn by a trained technician. Amplification was carried out using 2 large overlapping amplicons, and sequencing was performed in Illumina HiSec 2500. And for analysis, we have carried out this following tools, and to analyze those filtered variants, we have used several mitochondrial genome databases and mutation prediction tools, which I have listed down here below. And coming to my results and discussions, first we analyzed the distribution of several variants in different complexes of mitochondrial genome, and found that the D-loop non-coding region is most altered, and ND6 gene is least altered. They have found that the D-loop region is 60 times more susceptible to mutation than other region of mitochondrial genome, and polymorphisms in this non-coding region plays a very important role because they contain sequences which are responsible for replication and transcription. Furthermore, studies have suggested that mitochondrial D-loop MN1 or 2 site mutation might be associated with increased breast cancer risk. From my analysis, I have found a total of 119 non-synonymous variants. Out of that, 53 were found to specifically in breast cancer samples. And these are the list of those non-synonymous variants which are found in breast cancer samples. The first column are the gene name, position of nucleotide change, and their subsequent amino acid change, their frequency, and SIFT and polyphen to predict whether the substitution is damaging, and so to give the state of hydrophobicity and hydrophilicity. And p-mut score gives whether the substitution either decrease or increase the protein stability. The red ones were predicted to be damaging as per the SIFT and polyphen. Also, we found one novel mutation, and the blue ones are those which has already been reported for breast cancer in other population. This is still a continuation of table 1. And here, out of those 119 non-synonymous, these are the remaining 66 non-synonymous variants that are found common in cases and control. Likewise, in this table, the ones that I've highlighted in red were predicted to be damaging, and the ones that I've highlighted in blue, they have already been reported for their association with breast cancer. The list goes on, and these are the ones that are found in the D-loop regions. So coming to the discussion part for this analysis, we found one novel variant in Mizo population, and ATP6A8860 gene was found in 100% of breast cancer tumor tissue, which suggests that this variant may increase breast cancer risk significantly. Additionally, patients with more non-synonymous variant in ATP6 gene had been suggested to have a shorter survival compared to those with only one variant. ND3A10398G has been a variant of interest, and it has been suggested to be a possible candidate marker for breast cancer development. And this particular variant was also found to be associated with breast cancer in several populations such as American, European American, Polish, Malay women, and so on. This substitution changes the amino acid threonine to alanine has been referred as a reliable biomarker. Tomasi et al. Also observed this frequency T1612C variant in hormone receptor negative compared to hormone receptor positive patient. The variants in the coding region which were predicted to be affecting the protein function might contribute to breast cancer susceptibility in the Mizo population. Triggered by the environmental factors, it is well known that mitochondrial DNA polymorphisms or different alleles of the same locus could act as a disease risk factor in certain haplogroup. And in here, we analyze the distribution of synonymous.
Conclude. Time is.
Okay. So coming to my conclusion, this study focused on the contribution of mitochondrial DNA alteration and their association with increased breast cancer risk. Is the first time to report for the population of Mizoram. And we conclude that mitochondrial DNA mutation does play a direct or indirect role for breast cancer progression, and several variants that have been reported for other diseases might also contribute for the development of breast cancer, since there is variation in the risk dependent of haplogroup that is determined by the mitochondrial DNA polymorphisms or difference in allele frequencies aided by environmental factors. However, a study with more sample size is required in order to establish the extent of their contribution in breast cancer occurrence. Thank you.
Thank you. Yeah, next speaker, PC Lalolo. Behavioral and gene alterations in autism spectrum.
Good evening, everyone. So today, my name is PC Lalolo, and I will be presenting on the topic behavioral and gene alterations in autism spectrum disorder.
See this side and no, that one. Good. Your pointer should be this side.
What is that?
Yeah. Yeah.
Yeah, to begin, let me explain what autism is. So there are some individuals who, when they get upset, they make repetitive movements, and when get angry, they would bang their heads to the wall, and then even it would end up in bleeding their heads, and it would require stitching. And there would be some individuals who had talented gifts, where they would have great, remarkable gifts in mathematics or music, and also they might, when trying to make social interactions, they might struggle with making interactions, communications. So all comes under the autism spectrum disorder diagnosis. So here are some of the diseases involved in autism spectrum disorder. So here is a prevalence as of 2023. So 168 million people are affected worldwide, globally, and we have this starting from here. 4:1 male is female ratio, and male are more affected than females. And starting from 1970 to 2023, so there's 1:36 increase. So that is about 317% increase since 2000. So here are the countries that are leading the cases and the prevalence in the worldwide. So here are the common signs and symptoms like repetitive behavior, poor eye contact, hyperactive tiptoeing, poor social and communication, sensitive to light, sound, and smell, and delayed development in speech and learning. So here are the related disorders: Asperger's, Fragile X, intellectual disability, and global developmental delay. So what causes autism? So firstly, the main thing is the father's age during the time of conceiving. And the second thing would be the prenatal condition during of the mother. And the third thing would be the drugs taken during the mother's conceiving. And also the fourth would be the heavy metal exposure and genetic factors. And we are not sure on what mainly the cause of autism is, but one thing I can show off is that vaccines do not cause autism. And I will be focusing on the genetic factors, not because it is a sole cause of autism, but mainly because it is a field and that we can readily define and also better understand how the brain works, and also provide strategies to implement. So how genetic is autism spectrum disorder? As of 2023, 800 genes have been identified, and compared to other diseases, this is much more genetic than the other disease compared to cancers and diabetes and so on. So there are genetic conditions where they have discovered powerful genes and a single factor which determine this effect. And also there are 75% still unidentified, and also they might be contributing to the cause of that disease. So these are the commonly mutated genes that are reported globally. And these are the methodologies. First is a questionnaire through ASD DSM-5, and there will be asking the child's development, present history, and also their parents' demographic, parental history, and prenatal conditions. And these are the methodologies that we are implementing. And these are the 4 tests made mainly whole exome sequencing, metagenome sequencing, toxic metal testing, and melatonin ELISA assay. But today, I'll be focusing and represent results on whole exome sequencing. So here, the study site is a Child Developmental Center, where in the picture here we can see that the kid is being given occupational therapy, where his motor skills and attentional skills are trained. And currently in Child Developmental Center in ISO, there are 45 ASD patients currently. And these are the target sample size. And these are the results. The first result that I want to present is the genetic distribution. That is, from our sample, we have 77.1% male and 22.9% female, indicating with the other results that have been found out that male are more affected. And from the questionnaire, parents have claimed that 65.7% have psychological disorders, and the mean age is 5.5 years old. And in our study, we have 17.1% twins, and from that twins, there are 18.6% has autistic twins. And here, from the mothers and the fathers' age during conceiving, so usually from 31 to 40 years of age, they would have the baby which are autistic, and from there itself, proving the research papers which have already been identified that between 31 and 40 years of age, fathers are usually affected, and the kids would also be eventually affected. So these are the developmental histories, and here we can see that usually the kids are second child, and also the mother had a miscarriage. 29% had a miscarriage, and also this valproic acid is a disease. Is a drug that is taken during epilepsy. So these are the dietary habits, where like most of them doesn't like meat, they take meat at all. And these are the gene variants from the whole exome sequence that we could find and filter out based on the allelic frequency and the genetic mutations. And to conclude, there are still many things that we don't know. And also, I wanted to end by saying that also creating an awareness that autism people should not be, or individuals should not be treated as mentally challenged individuals, but their brain wires differently, and they think and process things differently. Thank you.
Thank you so much.
Good evening to everyone. I'm going to talk about the impacts of diacetyl hyperestrogen on women health, beneficial or harmful. As we have seen, Professor Jagatkanta has already talked about phytoestrogen. However, many of us were not here, so I would like to repeat it. About the concepts about the phytoestrogen. Phytoestrogens are naturally occurring non-steroidal phenolic plant compounds, and their structure resembles with the vertebrate steroid estrogens. And it exerts estrogenic, or it may exert estrogenic or anti-estrogenic effects in individual. The amount of phytoestrogen produced by a plant depends mainly on the growing condition and the plant cultivator. There are different sources of phytoestrogen found in a food like soy, like many other legumes, fruits, like red clover. So there are varieties of foods we use as a dietary supplement and a daily basis food. So here we see there are different types of phytoestrogen, mainly isoflavones, comestans, and lignans. Out of them, the isoflavones are the most active, most studied isoflavones, such as genistein, diazinein, glycitein, formamantine, and bioquinin A, found in soybeans and other legumes, including in red clover. So these are the biological effects sir has already mentioned about the role of phytoestrogen in human health and other diseases. So here we can see the phytoestrogens are promising candidate for chemoprevention and chemotherapy, and it have a very protective role on atopic dermatitis by upregulating the gene expression of the protein coupled estrogen receptor. It has also have shown that phytoestrogen improved oxidative stress in diabetic patients. Furthermore, phytoestrogen such as hormone inhibit inflammation and promote gastric mucosa via regulating NF-kB signaling pathway. And moreover, it has also impact on lipid profile and on the anti-aging. It also acts as anti-aging on the skin via estrogen receptors. So here are some more phytoestrogen enhances bone formation and increases bone mineral density. It also is a promising compound for the metabolism improvement and treatment of metabolic disorders. Genistein is also one of the phytosteroids, has been used for treatment of immune disease in animal model. It has been found that genistein inhibits allergic inflammatory responses. Another report also shows soy isoflavones for prevention of colon, endometrial and ovarian cancer. On the other hand, exposition of women to phytoestrogen, especially isoflavones, ligands and components of different botanical sources, it protects pre and post-menopausal period, and it helps the symptoms caused by menopausal like hot flashes, vasomotor symptoms, vaginal atrophy. So we have seen here phytoestrogen have different effect on lipid, gastroprotective role, anti-dysmenorrheal, bone loss, anti-aging, menopause, anti-cancer, and so many. So, but we have to understand that this is a one side of phytoestrogen. There are another side of phytoestrogen. Why it is? Because dietary phytoestrogens are bioactive compounds with estrogenic activity, and with the growing popularity of plant-based diet, the intake of phytoestrogen legumes, especially soy, have increased. The average daily intake of community phytoestrogens in East and South East Asia is estimated to be between 20 to 50 mg per day, which is sufficient to cause endocrine alterations. Okay, so I am going to skip the content here, but here I have to say it is considered as endocrine disruptor due to its serious health issues on human and other animals. So this is the one of the phytosteroid I have published in last year. This is a diazoline found in a fenugreek. This is also has considered as a phytoestrogen, and we have seen here it has interrupts metabolism, physiology, and reproduction of cis albino mice. So here we have seen there are alterations in these slides. So this figure here, so the steroidogenic alteration, 3-beta HSD, LHR receptors, and here you can see these are the aromatase receptors, and these are the testes and ovary histology
Come to conclusion.
chemical structures, and we can see alteration is there. Okay, I'm going to skip these all. These are the uterus of the mice showing the endocrine effect and reproductive toxic effects. So, okay, this final outcome I have to talk. So, phytoestrogen have beneficial effect, but it is also considered as endocrine disruptor by oxidative stress, DNA damage, hormonal impairments, inflammation and apoptosis, and reproductive abnormalities. So, on the basis of these concepts, we can conclude that intake of phytoestrogens on daily basis does have some physiological and metabolic effects that may influence the reproductive health of individual, and this effect depend on the factors such as dose, duration, types of compounds, bioavailability, endogenous hormone level, and health status of consumers.
These are the references, and thank you.
Thank you. Thanks.
Hello everyone, my name is Sarah Fawzwani. I will be presenting on oral microbiome composition among substance use disorder patients in Mizoram population. So substance use disorder is a chronic relapsing disorder which is characterized by compulsive behavior, and from Dr. Ashok Verma's presentation that we know that HNSCC 75% is caused by use of alcohol and tobacco. And in a study by NDDC, we find that opioid use in Mizoram population is 6.9%, which is higher than the average in India. And this data of Mizoram data is higher than the national average. Then SUD directly or indirectly affects our oral microbiome, which has around 700 species, which produce essential nutrients and amino acids. Due to time constraints, I will not go through the methodology. They will be clear in the results section. So I will skip over them.
Sure.
So moving to results. So in the social demographic characteristics, we find that there are no significant difference in the age and the sex between SUD and control, substance use disorder and control. And when we plot a Venn diagram of the species composition between the cases and control, we find that SUD patients have more unique species. Then when we do a diversity plot, we find that cases and control vary a lot in the species composition at the phylum level. So we can see from the coloring itself. Then when we plot the alpha diversity, we use 4 indices observed for richness, KO1 for O2 abundance, Shannon for richness and evenness, and Simpson for diversity. We find that the alpha diversity between the cases and controls are significant in all 4 indices. Moving to beta diversity, we use two indices. In weighted unifrac, we see the horizontal distribution, which signifies that the difference between the cases and the controls is due to differences in unique taxa. And in Brachytis, we see this vertical orientation, and in the cases, we see a more spread out points, which means that cases have more difference in the samples as well, which is evident in the microbial network plot, where in the controls we see that the points are clustered together, which means that the microbial composition is homogeneous in nature. To see which of these taxa have the most effect on that diversity, we plot the top 10 taxa, and we find that 2 of which are not significant and 2 of which have no higher levels of classification. From the 6 that are remaining, we find that 4 of them are higher in control, whereas 2 of them are higher in cases. Then moving on to differential abundance, using LIPC analysis, we find 47 genus level taxa which have the most effect on the diversity. And 20 of these taxa are from cases, and which all belong to the phylum Actinobacteriota and Firmicutes, whereas 27 of the taxa that were seen in the control are from Bacteriota, Firmicutes, and Proteobacteria. Then we moved on to functional prediction, and after filtering, we have 31 pathways that were annotated and identified. We have 6 major pathway classes. In human diseases, we have 2 pathways, and in genetic information processing, we have 5 pathways, and in lipid and amino acid metabolisms, we have 7 pathways, and in carbohydrate metabolism, we have 2 pathways. All these pathways are significant in a mean proportion level of difference of 0.1%. Then we have this PCA plot of the KAGG pathway, and we see that the pathways also differ between the cases and control. When we run an LDX2 analysis, which is for differential abundance in the KAGG pathways, we see that spliceomes and proteosomes have the highest effect. And when we adjust the p-values less than 0.05 are considered significant. So to conclude, we find prominent differences between the alpha diversity and the beta diversity, as well as the pathway abundances. This shows that a microbiome have a potential impact, and we need to further validate this with larger cohorts. These are my references.
Thank you.
And my acknowledgements. Thank you.
Still.
Good afternoon. My name is Tejavitso Chase. I'm from Mizoram University, and today I'll be sharing about the dietary niche preference. niche differentiation in 3 inaugural species in Kohima district. So in Kohima district, terrace paddy cultivation and zoom are like 2 major methods of agriculture, and there are over this is not in one part from like different sources. There are about 50 species of anurans reported in Nagaland. So these species they occur, as we know, like amphibians. They occur in and around water bodies, wherever there are moisture, wherever there is streams or ponds. They also occur in paddy fields, especially in the paddy fields in Nagaland where we have this embankments. So they also occur there. And whenever there is one or more species, there's always some kind of niche overlap. So they share resources in terms of maybe it's in terms of food, or in terms of space, or in terms of their the oral niche. So both the theory of natural selection and competitive exclusion theory suggests that the only species that survive in an environment is the strong one. The weaker ones get eliminated; they go extinct. However, according to Bianca 1974, that is not always the case. That did not always be the case, because species can also co-occur in the same place at the same time. So for this work, I surveyed paddy fields from 5 villages in Kohima district, and I used a notable visual encounter survey. Gut contents were extracted using the following Solar et al. 2005. So also I selected 3 species. Those were Euphlyctis, Cynophyctis, Polypedids himalayensis, and then the Minervaria nephalensis. So for the gut content extraction, first I hold the frog with one hand and then open the mouth with forceps, and then inject water from the habitat into the stomach of the individual. And then when the water is pushed into it, the water comes back out, and then the frog also creates this regurgitating effect on the frog, and then it regurgitates the water and the food together. So the food from there are stored in 70% alcohol, ethanol. So volume of prey, these are some of the formulas I used. Index of relative importance was used to see which prey is most important to the test. And okay. So in this survey, the vacuity index was pretty high. Many of the species had a high percentage of the individuals had empty stomachs, and even the ones which had food in the stomachs had very less amount of food in the stomachs. So on average, for Euphlyctis and Anophyctis, there were only 3 individuals per 3 food items in the stomach, and for Minervaria nephalensis, there were only 1.8 prey item in the stomach, and for Himalayensis, Polypedids himalayensis, there were only about 2.9. So even previous studies have all for dietary generalist species. Those are species which can feed on a variety of prey items. In generalist species, we'll usually see that coleopterans, or those are the beetles, are the maxima. And similarly, in this study also, the most common prey items were the coleopterans. These are insect arthropod prey orders. And besides Coleoptera, Araneae, which are spiders, and also Diptera, and okay. Yeah, so again, the least breadth was widest in M. nephalensis, and then followed by E. Cynophyctis, and then P. himalayensis. Niche overlap was highest in between the Polypedids himalayensis and Nephalensis, Minervaria nephalensis, and then followed by P. himalayensis and Euophyllus, Cynophyllus. And then lastly, between E. Cynophyllus and Minervia nepalensis. So the high vacuity, the reason for so much empty stomachs or very few prey item in the stomach could be because of the sampling period, which was done towards starting from the dry season of the year into the monsoon. But because of the sampling in the dry season, it could be less. Also, similar to other studies, coleopterans were found to be the most important prey item. There also, I also observed high niche overlap between the species. But then there was one point to be noted is that their microhabitats were different. Yeah. So that is all. Thank you.
Thank you.
Myself, Kitiko Satnath. I'm PhD research scholar in Department of Zoology. My guide is Professor S. T. Liam Sanga, and I also working in genocide inaugural. My topic is amphibians of inaugural wildlife sanctuary, diversity, distribution, and their conservation. As we all know, amphibians are cold-blooded vertebrate, and it's play a vital role in cleansing ecosystem. It's least harmful to human. And in India, there is 457 species till now, and in Arunachal Pradesh, in 102 amphibia. And my study areas is mentioned in title is Eternal Wildlife Sanctuary, which is about 140 square kilometers. This is the map of Eternal Wildlife Sanctuary. Then data collection, I did my all the study from November 2019 to September 2022. Amphibians are collected through foot and trails by adopting active searching method, and roadkill data also I gathered, and these are species identification with the help of these literatures. Then data analysis and land use land cover data analysis by using QGIS, and diversity index and study analysis are performed by using Excel and PASS software. So my result, till now I collected 162 individuals of frogs and toads, from which I found 32 species belonging to 18 genera and 6 families. Out of these, family Dicroclecidae found highest in composition of species, while it's minimal in Megaphilidae. Only one species I found. Among the genera Minervaria is found highest, followed by this. These all are the genus list with the species, and this is the pie chart. Then this some of the pictures of amphibians. Then this land use land classification. This the classes of habitat. While interestingly, that built up area, this red portion of this map, built up area is 25% now, which is the result of encroachment. This is the diversity index. In forest land, we found diversity highest. Xenon index I found 1.859, and one is minimum in cropland. This is threat to amphibians of Arunachal, this Eternal Wildlife Sanctuary, hunting for consumption, roadkill, and habitat destruction. This from the questionnaire data, we found that 89% of respondent goes for hunting, while 11% of respondent is do not have. For this 89%, 92% of that respondent is hunt for only for food, and other 8% is for local trade, and both the food and local trade. This summary is, and this habitat destruction, as I mentioned in LULC map, 25% of total land is now encroached. This is all the encroachment photos. Then roadkill threats. I did 2 years of roadkill survey, and I found that monsoon, pre-monsoon and monsoon season is peak season for the roadkill, and this when it is minimal during winter season. For the discussion, this is the first ever survey of amphibians in Arunachal Pradesh. Though it is this Eternal Wildlife Sanctuary is situated in heart of the state, still this habitational study is neglected for this period. And anthropogenic activities leads to decline of population and leads to climate change also. It is also result of emerging disease. As you see, this all the abnormalities found during the study. I sincerely acknowledge my professors, PCCF. Thank you. Thank you. Thank you. Thank you. Hey.
Good afternoon, everyone. My name is Lel Mosang. I am doing my PhD in the Department of Zoology, MZU, under my supervisor, Dr. Mizoram Sanga. Today, I will be presenting on the topic of biology of Nagaland Mountain Torrento to the finance standard. So, let me skip all this and go to results because, okay, to study this distribution, habitat, and morphology of the finance standard in Mizoram. The distribution was studied using by using the visual encounter system and acoustic encounter survey and all. You can see the maps here. So it's all highlighted here. I will go next. Morphology. These also finance standard were medium-sized toads. Female toads had a mean SCVL of 62.89. Let me go to this. These are my morphologic data, morphometric data. I will go directly. These are my study sites at Moorland National Park. This is the vegetation. The vegetation, according to Champion and Set, we have identified several species of plants around the study sites. So these are some of the, and this is the temperature, humidity, and pH graph throughout the year, maximum and minimum. And this is the calculation for slopeness to all of my study sites. So to analyze phylogenetic data, phylogenetic data is already published in 2020 by H.L.M. Sanga et al. So we compared the data with that data was acquired from Huifang Community Reserve Forest. So we compared the data from Moorland, and it is found to have a non-significant 0% divergence from of Huifang and Moorland species. So histological studies was conducted with the help of Sarvigas, and we have characterized all these things. Breeding biology, breeding season. This frog is very amazing. It breeds only 4 nights in one stream per year. So it is very hard to get them in the field. We have to stay in the field at Moorland National Park. And though we have identified more than four distributional places in Aizawl, we have only located only 1 breeding site in Mizoram, since the breeding season lasts very short. That is only for around 4 nights. Ovipositioning, breeding behavior, ovipositioning, and all. So you can see my study sites here. Even multiple lemplexas you've seen. So while studying the developmental biology, we have to transport the tadpoles, the eggs to Mizoram in the laboratory. So that is done using an oxygenator and with the help of inverter and batteries. So even acoustic analysis was also done using Raven Pro. Raven Pro is a software developed by the Californian Bird Organization. So parasite was extracted from intestine of the preserved specimen, and it was sequenced using 12S gene. And after we blast it, we found to be from the genus Cosmosarcoides. And this is the study of the embryonic and larval development. So this is the table. So you can see from Great Crescent to upper column development is 19 days, 22 hours. And here I label this in dates. So from Gosner stage 1 to Gosner stage 38, it takes around 58 days. Effect of hypoxia. Developing embryos need minimal supply of oxygen, dissolved oxygen. So if there is ample amount of oxygen in the breeding sites, there will be some deformities in the development of the tadpoles and even the eggs. So this was published in Heart Bulletin in 2022. This is to study the oral morphology. LTRF, labial 2 draw formula, of course, now stage 27 and 31 is 2/3. So these are some, since this toad is breeding in very high elevation, there are less diversity in plankton species also. So we find only Gomphonemma, Sphyrogyra, Penularia, and these are the diets of the adult species. Here, the picture on the so the diet mainly consists of Coleopterans, Hemipterans, Orthopterans, Hymenopterans, and from Arania. This is spiders. And this is my conclusion. Though thus it are recorded male toads to have SVL more than 60, the study shows that there was huge gap in SVL of male and female. The breeding behavior of the species were thoroughly studied. The breeding season tends to start from late February and ends mid of March. This should be beginning of March. This is where the species is found to be an explosive breeder. The genetic distance with the closest species to Taphida swarti is 3%. Since there was no genetic data from the whole type species, this is the first study on genetic information. Parasitation was observed. The investigation of gut content of tadpoles shows that the high elevated lotic streams have fewer planktons, as in Islam 2021. The tadpoles were highly opportunistic feeders, feeding on leaf, dead insects, and detritus. Adult food studies show that the species were carnivorous, feeding on insects of different kinds. This concluded the species are opportunistic carnivores. So, the main reason why I present this presentation is that I appear on this program also is that what us as a field researcher can do to the community. So we have conducted several awareness campaigns, more than 17 in different parts of Mizoram last year. Plus, this is the last point. This is a homestay in Moorland. I, along with Amit and the VC secretary, we have set up this homestay there at Moorland, so that research scholars from all over the world, all over the world can stay there and do the research there, and this is of minimum cost. So we have done it personally from our pocket, Amit and me. So what I want to conclude is that let's work out for the community as we are pursuing on our research. Thank you.
Thank you. Thank you.
Good evening, everyone. Myself, Martin Puya. I'm from Department of Biotechnology, MSc. My topic is inventory survey for fruit piercing moth Kalpine in Mizoram University campus. Introduction. Kalpine is one of the subfamily of fruit piercing moth in the family of Erepidae. Fruit piercing moth are serious pairs of fruit crops throughout tropical and subtropical, belt from Africa to Southeast Asia and Australia Pacific Ocean. Most of the Kalpine are serious adult pairs of replants fruit for variants of Pomiosia fruit. Example, as you can see, such as citrus and etc. Unlike other moths, the dwarf moth is feeding habitat is destructive, while the larvae are not. And the larvae feed only in massive spermacia, and egg of these moths are laying coral. And this is a taxonomic person of the subfamily Kalpine, and within this subfamily Kalpine, we have three types, namely Kalpine ophiderinae and Phalloderinae. Morphological feature. The subfamily includes many species of moths that have point and barb proposis adapt to piercing the skin of fruit to feed on juice, and in case of several calyptra of bompier moth to pierce in the skin of umbel to feed on their blood. And this is the picture that I put here is Eugosima homoinea. Most Kalpine have orange abdomen, as you can see in the picture. Forewing are usually dull in color, and high wing are usually orange, and dark spot can be seen in the high wing. The proximal region of the proboscis is simple and smooth with circular and semicircular ribs. Proboscis are usually well developed and extremely long in some species that feed on flower in deep calyx. A dual rib feed on ribs and animal tears or blood. And I will material method. I will simply tell about that. This is a light trap that we set, and we collect using this light trap and specimen are like hand sample and kill it. The killing jar that contain petroleum ether. Hence, most are nocturnal insect survival was only conducted at night by searching from building, hostel, department, etc., and any light source that can insect may get at their coast with the help of net. And identification was based on the river literature. Virtual of collection specimens were maintained in systematic and taxonomic laboratory, Department of Biotechnology, Mizoram University. Genital dissection was performed, followed by Lee and Brown. Photograph was taken using multicam microscope. Result: to provide study the first documented carpini from Mizoram. Hence, all species recorded during this study period is the first new records for carpini not from 9 Mizoram. Total of 14 species were collected during this study time, and 5 species belong to 3 genera. The majority of the specimens that we collect are female. And this is the checklist of the carpini that we collect used inside Mizoram University campus. And this is the picture that we taken. And this is male genitalia, and female genitalia. And these are the proboscis of the fruit person mode carpini. And as you can see, there is a serotypes in the tips of the proboscis, and these are the usually that feed on the juice of the fruit. And my conclusion and my last slide, this is extensive survey of literature, report on the diversity of carpini in Mizoram. Therefore, there is no urgent need for the scientific document on us explore fruit piercing mode of the Mizoram. The study will be one step forward, increase knowledge to research the biodiversity of the state and country in general and fruit piercing mode in particularly. Thank you.
Well, thank you so much.